In this video, we will discuss about immune pathogenesis of SARS-CoV-2. So how this SARS-CoV-2 when enters the host, how it will activate the immune cells and how the immune systems will try to eliminate this SARS-CoV-2 virus. So it starts with this SARS-CoV-2 when it interacts with the ACE2 receptors and it enters the cells of the host, and where since this SARS-CoV-2 virus contains genetic material as a RNA, this virus replications occurs, or in other terms, a part of the virus will replicate it and part of the virus is converted into the proteins that is seen here, the replications part and the protein part, and after that some part of this viral protein will assemble together in the Golgi apparatus and its release. And when it reaches to the other cells, for example in the case of lungs, then because of the degradation of the SARS-CoV-2 virus in the form of these PAMs and DAMs, then it will be detected by the macrophages as this foreign harmful particles, and this will activate the macrophages, and that leads to the release of the chemokines and cytokines. And also these activated macrophages will continue to release these chemokines and cytokines in the extracellular matrix. And the next part, what will happen is that the activated macrophages, when it releases the chemokines, not only in the extracellular matrix but also within the cells, and this will lead to the macrophages will lead this portion is the zoom in view of this lung portion. So when these PAMs and DAMs detected by the macrophages, it gets activated and release the chemokines and cytokines that will activate and trigger the other immune cells to come at the site of infection. At the same time, these chemokines such as IL-16 will activate the T cells and B cells and helps in the differentiation of B cells into these antibodies against those PAMs and DAMs. And other thing that this macrophages does is it will also activate this neutrophil 3. It will also activate this neutrophil, not neutrophil 3. It will also activate this neutrophil through these cytokines such as interleukin 1 beta, and the activated neutrophils undergo degranulations, and that helps in release of the chemokines and cytokines at a site where the PAMs and DAMs are not yet reached, and this will help in the increase in the efficiency of the immune response against the SARS-CoV-2 virus. And the next thing that had happened, this dendritic cells is about this dendritic cells, since now the cytokines are in the extracellular matrix also. So these dendritic cells have the informations about these PAMs and DAMs, or non-specific harmful antigens, and these dendritic cells will use those informations to activate these T cells with the help of this cytokine release, and also with the help of this major histocompatibility complex 2 that will help in providing the informations of this non-self harmful epitope to this activated T cells. And the other part that occurs in this panel B is about this activated macrophages will also attract other immune cells at the site of infections. That can be seen here with the help of release of this cytokine storm such as interleukin 1 beta, IFN1, CXCL10, CXCL11, interleukin 6, IP10, and TNF alpha. So all this cytokine storm helps in recruiting other immune cells at the site of infection. So other things, this SARS-CoV-2 virus, when it in order to trigger this immune response, the SARS-CoV-2 virus, when interact with these endothelial cells, then the activated T cells are present. When the new SARS-CoV-2 virus is trying to enter the endothelial cells and the activated antibodies against the SARS-CoV-2 are present, it will trigger immune response through different pathways such as the C1q pathway, that is classical pathways, or the lectin pathways, and both of them leads to this C3a and C3b or both of them converges to this C3A and C3B, and after that they will leads to this C3A and C3B leads to the formation of C5A and C5B. So in this case, C5A, the function of C5A is to recruit more immune cells such as neutrophil monocytes. It is, however, the function of C5B is to form a membrane attacks complex, and that will help in lysing the cells that are infected with SARS-CoV-2 virus. And this membrane attack complex, that will in this case what will happen is that this membrane attack complex will help in lysing the cells, and that might even leads to lung damage. That has been shown here. So this is how the immunopathogenesis of SARS-CoV-2 virus in briefly explained. If you need to understand in detail about this immunopathogenesis of SARS-CoV-2 virus, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.